Hello everyone, welcome to my channel Instant Microbiology and Biotechnology. Today we will discuss about the principle of biodegradation of pesticides and xenobiotics and why we need to degrade the pesticides or the xenobiotics. So let's get started. So come to the introduction. Pesticides is basically a mixture of few chemicals that may prevent or mitigate or kill or repel the pest or different type of fungus or different type of bacteria, etc. For example, insecticide that kill the insect, herbicides that kill the unnecessary plants that has been evolved during the farming process. And third is fungicides, basically some fungus that kill the plant or that cause the disease in the plant. So in order to control that, we use this fungicides. So mostly these are called as xenobiotics, or these are certain types of xenobiotics. Now the question is, what is xenobiotic? Xeno means foreign, biotic means toxic substances. So basically, a foreign chemical that is toxic in nature, which cannot be produced biosynthetically or chemically, which are man-made and do not occur naturally, is called as xenobiotic. For example, is aromatic, pesticides, certain hydrocarbons, plastics, DDT, many more. So what are the properties? Basically, these are chemically synthesized products and toxic in nature. They are highly stable and chemically, biologically inert due to presence of substitution groups like halogens, nitrogen, sulfate, amino or methoxy or carbamyl groups. Though in 1975, total 1170 number of pesticides were registered for the use, including 425 herbicide and 410 fungicide and 335 insecticide. Among more, but most of the agents are chlorinated pesticides. Mostly these are resistance to any type of degradation, especially the microbial degradation means biodegradation. Okay. Hence, the excess use of pesticides creates a imbalance in the environment because we cannot biodegrade it easily. So what are the imbalance? So it causes environmental pollution, soil pollution, include imbalances in the nutrients that is present in the soil, and presence of microorganisms in the root or also get disrupted, means the disbalance has been seen in the root section where the soil is associated with it. Among these pesticides, some are biodegradable, some are non-biodegradable. Okay, if something is biodegradable, we should know the principle of biodegradation. So according to International Union of Pure and Applied Chemistry, biodegradation is breakdown of substances that catalyzed by the enzyme in vitro or in vivo. The ability of microorganisms to convert a toxic chemicals means some xenobiotics to simpler or non-toxic compound by synthesizing certain enzymes or certain metabolites. Biodegradation process affected by substrate specificity, nutrition source, temperature, pressure, pH, etc. The xenobiotic compound can be degraded by two different ways. One is oxic, that is presence of oxygen, and anoxic way, that is absent of oxygen. So in oxic condition, some species, for example, Pseudomonas, Microbacterium, Nocardia, algaligens, yeast, molds, are involved in the oxic degradation of xenobiotics. Sorry, I'll make it capital. This P I have mistakenly made it small. So for example, some aliphatic moiety that converted to alcohol, then alcohol converted to aldehyde, carboxylic acid, then it enters to fatty acid cycle, from where it can easily enter to the acetyl CoA through beta oxidation, and then it entered to TCA cycle. When it entered to TCA cycle, the things is easier now. It can be degraded easily. Similarly, in aromatic moiety, it is degraded to produce catechol and then to degrade to dicarboxylic acid. Now this dicarboxylic acid through beta oxidation produces acetyl CoA and then it enters to TCA cycle. Similarly, in anoxic condition or in absence of oxygen, microbes like these denitrifiers, sulfate reducing bacteria, desulfomonas involved in anoxic degradation of xenobiotics. Means these organisms can degrade in absence of oxygen. In anoxic degradation, in place of oxygen, these microbes, means these denitrifiers, sulfate reducing bacteria, this utilize nitrate, sulfate, ferric ion as an electron acceptor. Generally, reductive dechlorination occurs in anoxic degradation, especially by desulfomonas. Means the longer chlorinated chain degrade gradually and forms a certain molecules. Then we should know what are the different methods that has been used or that has been followed during oxic or anoxic way of degradation. So first is detoxification, means conversion of the pesticides to non-toxic compound, means toxic to non-toxic. A single moiety in the side chain or removal can render the chemical to non-toxic form. Okay, then second is degradation, means breakdown or transformation of complex substrate into simpler product leading to mineralization. For example, trium is a fungicide is degraded by a strain of pseudomonas and degrade the product and form dimethylamine, proteins, sulfolipids, etc. Then conjugation means formation of a complex or addition of that particular pesticides or herbicide to the metabolite. So basically an organism makes the substrate more complex or combines the pesticides with the cell metabolites. For example, amino acid, organic acid, methyl ground or certain substrate, thereby interacting with the pesticides. Means these metabolites will interact with the pesticide and form a conjugation. So then it can be easily accessible to the microbes for the degradation. Then fourth one is changing the spectrum of toxicity. Means if a certain molecule is targeting to certain type of organism, it can change the spectrum. Means by the certain molecules, suppose certain pesticides, it designed to control a particular group of pest. However, when it interacts with other substances like with the soil, it get mutated and inhibit to entirely dissimilar group of organisms. For example, a fungicide named as PCNB, it is converted to a chlorinated benzoic acid that kill the plants. Instead of only controlling the pest, it can kill the plants also. So these are the methods how we can degrade it. Now come to the degradation of pesticides. As I have mentioned before, there are different way of degradation like oxic, anoxic, and different method of degradation, but some can be degraded totally. Means we can totally make a non-toxic substances to toxic substances to non-toxic form. But some pesticides and some xenobiotics are there which cannot be degraded totally. Means we can just make the toxic form, the level of toxicity, to a lower level of toxicity. Means we can reduce the toxic amount, we can reduce the toxicity, but totally we cannot degrade it. And such molecules are called as recalcitrant. And what are the examples of the pesticides that can degrade easily? That example is 2,4-dichlorophenoxyacetic acid, 2,4,5-trichlorophenoxyacetic acid, and some molecules which are called as recalcitrant, means which cannot be degraded easily. The examples are PCE, that is perchloroethane, and DDT, that is 111-trichlorobase phenylethane. So these are the certain examples which is clearly indicating one is one can be totally degraded, and some molecules are still there. We cannot degrade totally, but we can lesser down, or we can shorten, or we can make the more toxic product to less toxic product. So in my next video, I will describe about the pathway, how this molecules degraded, and how microorganisms are producing certain enzymes and certain gene, how it is activating to degrade the molecules. So if you like my channel and you want to learn more, so please subscribe my channel Instant Microbiology and Biotechnology. I hope you all like my channel, and hope I will see you in the next video. Stay tuned with me. Thank you.